IEEE Young Professionals, as we know that we represent the young engineers, particularly the young electrical engineers of the country. So we also want to listen to a young biotechnologist who's working on the field and to know how we young engineers and young biotechnologists can collaborate to solve these problems. On that note, our third speaker is Dr. Mustak Ibn Ayub. Dr. Ayub is currently working as an assistant professor in the Department of Genetic Engineering and Biotechnology in the University of Dhaka. Dr. Ayub completed his PhD in molecular oncology cancer in the Cancer and Immunology Lab of John Radcliffe Hospital, University of Oxford. He's the director of Network for Young Biotechnologists of Bangladesh. He will talk about generate, complete, interpret, and integrate, a data-centric model towards interdisciplinary research. So let's hear from Dr. Ayub.
Okay, thank you so much. I'm really excited to be present in this platform. I don't have many experience of talking with engineers, but this is a really good opportunity to share some of the work that I did and also some of the thoughts with you, the way we can collaborate more to solve not only just this problem, but also some of the persistent problems such as cancer research. So the title of the research of the presentation is Generate, Compile, Interpret, and Integrate. I'm basically trying to emphasize on the data that we have generated so far and how we can compile this data and integrate and interpret the data in the light of existing knowledge. And then at the end, we can integrate the knowledge, the distilled knowledge in the broader field. So I think I really should thank Dr. Ashraful Alam because he kind of made some of the things easier for me. Before I come back to the COVID-19 case, I want to emphasize on a paper that has shaped the field of cancer research. As Sajid was saying that I did my PhD in molecular oncology, and also Dr. Ashraful Alam was discussing some points about cancer. It is a disease which remains a major challenge for everyone, for every country around the world. But before 2000, the field of cancer research was really, I would say like, it was not disorganized, but it was really spread all over the places. And we didn't have a very concise and structured picture of cancer research and the data and the results that different researchers were bringing out. So in 2000, exactly in 2000, in the beginning of this century, two scientists, Douglas Hanahan and Robert A. Weinberg. Weinberg is a very popular biology teacher in MIT. They came up with a review paper. So in this paper, they didn't, as you can imagine in a review paper, they didn't present only their research, but what they did very successfully, they summarized all the works that were done in the field of cancer for last 20 years, 25 years. And using the data together, they were able to see what is the patterns which are consistent in cancer and what are the hallmarks of cancer that we can identify to say that some normal phenomenon in the body has been changed into a cancerous phase. And this became one of the most successful papers in biology field. And they also followed up their research in the paper in 2011 to celebrate the 10 years of the success of the paper. And this is basically the outcome of the research that they did. So from the elaborate revisit of the existing data, they interpret the data and then integrate all the knowledge and gave us the 6 hallmarks that can define the field of cancer or a cancer which is growing in our body. So I'm not going through the different components. It's basically talking about the various characteristics of cancer cells, like they grow really uncontrollably and they don't die or they don't want to die. They grow lots of blood vessels, etc. So this is a very good example how we can compile the existing data, and then we can come up with really revolutionary ideas in the field of biology. So this actually also was pivotal. The same principle was also pivotal when this new pandemic came. So SARS-CoV-2, the virus that we know at this moment as a reason for COVID-19 pandemic, when this virus first was identified in Wuhan in China, we didn't know that it should be called SARS-CoV-2. We knew that there is a new virus and some of the symptoms are like they cause pneumonia, they cause breathing problems in the patients, and the patients, sometimes the symptoms may be fatal for the patients. So we knew these things, but when they, when they looked at the virus under the microscope, obviously, and then the most important thing that they did is, as we have progressed very well with the sequencing technology, again, this is a field where electrical engineers and also other engineers can play a pivotal role to progress the field of sequencing technology. It has been revolutionized already. So when we sequenced this virus, it was called novel coronavirus. It was coronavirus, but we didn't exactly know what type of characteristics it has, what type of genome sequence it has. So genome is basically the sequence of the nucleotides that Dr. Ashraful Alam previously described. It's a RNA virus, so it has U, C, and G. So the genome means that these 4 nucleotides will be basically organized in different combinations. So, these are the building blocks. And in novel coronavirus, there were 30,000 nucleotide positions. So we knew these informations. And when we have this information in hand, what we immediately did was to compare this information with the existing viral genomes that we had in different databases. We have very structured biological databases in US, in European countries, in China, in Japan. So when we compared this information, we found that, well, it is a novel coronavirus 2019, but indeed it has a very similarity, high similarity with a virus that was common in 2003, which caused SARS, which was severe acute respiratory syndrome. And the virus was named as SARS coronavirus. So because of this similarity, so the sequencing technology gave us the sequence, and with the comparison with the existing knowledge, we understood that this is very similar to the SARS virus, and that's why we call it SARS-CoV-2. Oh. So it shows the power of the of generating data. So we are generating and then we are compiling the data, comparing the data, coming up with some conclusions. And this is even more important when we look at more predictions about the information that we have generated. So as I already have said, that SARS-CoV-2 has 30,000 and some of the computational tools already exist, and this is where the computing is a very powerful tool for us. And obviously, Professor Ashraful Alam has already mentioned the IEEE. Obviously, they have a big wing for computing. So using the computational knowledge, we can actually structures of novel coronavirus. When we compare the virus, the sequence with the existing is a very powerful way of using the existing knowledge to predict the future or to generate new knowledge when actually we didn't know anything about the new virus. But within very few months, we actually have known a lot of things because we could capitalize on the knowledge and the information that already we had in our hand. So this is Professor Zhang lab in University of Michigan. At the left corner, you can see lots of tools that they have developed, and this became very important when we wanted to look at the information that we are generating for novel coronavirus. I will come back to this the importance of these tools and how we can collaborate to develop more tools. So as the novel coronavirus became a sensation and the pandemic became like an issue, global issue of pandemic, like a threat for everyone, I also was personally encouraged and interested to have a look at the sequence information. And in April 16, I came up with a paper where I proposed that based when we analyze the sequence, like those ATCs, the 4 nucleotides, we can actually find some patterns. And based on the patterns, I proposed that we can divide all the coronaviruses in two categories. This is basically the mutations that I put emphasis on. In some viruses, not only this, at that particular time in April, I showed that the strains of these mutations, the strains that contains these mutations can also be correlated with the like the severity and the prevalence of SARS of COVID-19 in different countries. Just at the left, in this right graph, the first country is Spain. It has the lowest number of SARS-CoV-2A, I mean the mutated virus, but it had the highest number of cases at that time. And at the like at this end, in Portugal, at that time in April, we had highest number of this changed virus, like AAC mutated virus, and we had the lowest number of cases. Well, this observation is not valid anymore for all of these countries because the data size and the data has also become biased with different kind of contributions. But it was in predictions or assumptions that I could make in that particular time. And I think this shows us, I mean, I don't claim that this is really the real picture, probably it's not true, but actually it could gave us some assumptions and gave us some indications how we can look at the data, existing data, and use the computational tools and make some assumptions to shape the research in the research lab. Like for biologists like us, we can probably will be able to look at the mutated viruses and see whether they have actually reduced infectivity or like more enhanced infectivity. So this mutation analysis is a big part of biological research and predicting the impact of the mutations for the to monitor the course of the infections, the pandemic is very useful tool for us. So the changes that I showed in April, it actually elaborated much because at that particular time we had only 3,000 sequences. Now we have 50,000, more than 50,000 sequences from different countries. And as I said that the sequencing technology has been revolutionized, and because I think this is probably for the first time we have seen huge number of sequences within a very short period of time because of the advancement of the technology. So based on these 50,000 viruses, now we have been able to actually look at the more elaborate picture of the changes, and we have been able to divide the viruses in 5, we call them clades, in 6 clades. And in different clades, there are some mutations which are more prevalent or sometimes exclusive in different clades. Especially, for example, this S clades that you can see here, it was prevalent in the beginning, like as you can see here, but it decreased when the pandemic moved, like in May, in April, and also like in June. And we can see that these G, GH, and GR clades increased with time, and the S clade and the L clade and the other clade like this O, which we don't have like a proper definitions. So these clades decreased with time. Actually, what happened is in US and in North America, in South America, and in Europe, these viruses started to increase. So G, GH, and GR, it increased. And in China, as the viruses were really lowering with time, so we didn't see these viruses anymore in May or in April or in June. So this shows us how the sequencing technology is helping us to like to show the different variations, the different variants of the virus and how the viruses are moving with time, and not only with time, but also geographically. So here, just I'm showing the right bottom corner, the changes. So you can see in China, from this particular position, C to T, and we can reveal these informations through sequencing technologies in Europe and US, as I have already mentioned. And most importantly, with time, we can also see deletion mutations. That means that certain portions of the virus are now deleted in the genome. And this is very encouraging because this deletion means that the virus will be probably less pathogenic and less lethal. So with this information, scientists actually predicted a lot. For example, that particular mutation I mentioned here, if it's like this change, like D614, 614 positions in a protein, which is, I would say, which represents the RNA change in 23,403 positions from A to G. So this particular position, the scientists claim that, as already have said, that these variants are more prevalent in Europe and in US, and scientists have now claimed that these positions are, these changes in these particular positions have made the virus more infective. So that means that the virus can infect the cells more easily, and that means that it will spread from one person to another person more easily. And this has been predicted computationally, and this has now also been predicted in laboratory, like in real biological experimental setup. Based on the informations we also did, I collaborated with another student from Kulna University, and we wanted to see what is happening in the protein of the virus. The change that we see in this particular positions, like these 614 positions, where a D amino acid, these are the building blocks of the protein molecule. So D is aspartate, and this aspartate becomes glycine. And aspartate is an amino acid which has negative charge, and glycine is a neutral amino acid. So we wanted to see what will be the impact of the change. And interestingly, we can see that this change doesn't have any impact on the function of the protein. We try to use several algorithms, at least 3 has been mentioned here, and we found that this change is particularly tolerated. But we also saw that although this is the most prevalent changes, that means for more than 67% cases, this change occurs in the protein. That means in US and in Europe, this mutant virus is more prevalent. In 67% cases, we have this virus. But there are also some other minor viruses, minor variants, such as in 936 positions, we can find D2Y change. In 1263 positions, we can find from proline to lysine changes. And the algorithms predicts that these changes will have deleterious impact on the protein. That means that when this change will occur in the protein of the virus, this virus will be less effective and probably will have reduced infectivity. However, when we looked at the structure of the virus, like the 3D structure of the virus, so we did the simulation study, we did 3-dimensional analysis of the proteins, and you can see when in 614 positions, you have D, that is aspartate. It can make lots of bonds with other neighboring protein amino acids. But when you have G, which is glycine, it actually cannot make lots of bonds. That means that in this particular mutant variant, the protein will be more relaxed. And I'm not going to detail, but when the protein is more relaxed, it actually facilitates the fusion of the viral membrane with the host cell membrane. That means when we have D to G chains, it actually will facilitate the entrance of the virus into the human cell. So what we can do in the in the laboratory, in biological research, we can actually also do quite a lot by doing simulation studies, by predicting 3D structures of the proteins. And in these particular cases, we need the help of the people who are expert in computational biology, who are good at computing and come up with good algorithms and good softwares. So when we were doing this research, the challenge we faced, I'm talking in the context of Bangladesh. One of the main challenges, you have mentioned already some of the softwares that we used. One of the main challenges is actually the software is a black box to us. So the results that I get as a biologist, I don't have like in-depth understanding about the algorithms and also the programming languages. So I am completely dependent on the outcome of the results. I give the input and I look at the output. I don't know what happened in between. So I think it will be very useful if we can have some collaborations with a person who knows these things and can, it will also help us to probably change, to tweak some of the parameters, and that can give us different outcomes. I already have shown that not all algorithms agree with each other, and that is also another problem. We sometimes become confused which output we will depend on and which one we will reject. Another problem was, for example, we didn't have access to servers, we didn't have access to high-powered computing systems. So these can also be probably accessed through the collaborations with the people who are working in computing and also like other heavy-duty programmings. Lack of collaborations, as I mentioned, we have a lot of expertise, but those are compartmentalized. Professor Ashraful Alam mentioned that, well, a lot of things you probably will be able to pick from intermediate biology knowledge, but if you want to go deep, then you need to collaborate with others. So we need collaborations for that. And also another problem will be jargon-rich literatures. If I want to understand a topic, a biological tool or a computational tool that can help me to predict the biological data, it will be very difficult for me to capture everything. So what the solution will be, the main thing is collaborations, and for that we need to create an open ecosystem for frequent communications, more active research collaborations. I think COVID-19 has become an eye-opener for us, why we need more collaborations among different fields. We need probably informal research area. We don't always have to talk about serious research. Just casual mixing and casual sharing of research ideas can come up with big research projects. And at least it will help us to understand each other's strength. And then sharing resources. It will be very important to share the research with each other. My knowledge in molecular biology can be complemented with the knowledge of computing and also like sequencing technology, where I need the impact of electrical engineers. So I'm coming back to the last part of the talk. So everything that I have discussed so far, I think I can kind of conclude with these four components of the wheel, which are interconnected. One is obviously the biological researchers, they will generate data. As for COVID-19, the first data was the sequence, and then it has really led to many other things and kind of an explosion of various predictive research and also other lab-based research. So we need to whenever we generate the data, we need to compile the data. And I think at that particular time, we need very close collaborations with biology and computing people. We need to interpret the compiled data, and at that time we need biology, computing, and data scientists. Statisticians can be a great help. And also when we want to integrate the newly generated knowledge with the broader field, we again need the collaborations present there. And not only for this pandemic, as Professor Ashraful Alam has said, and we all know that it will not be here forever. It probably will be over in next 2 years or something. But there are many other changes, many other challenging fields where we can follow the same wheel, the same components, and collaborate, especially at this particular time. There are many data which are sitting idle and underappreciated on different websites, different databases. I really will invite the IEEE people to come up with the collaborative research ideas where we can collaborate to compile the data, we can interpret the data, and then integrate that to really have impactful outcomes the way the Robert Weinberg and Douglas Hanahan did in cancer research field. So this is basically my talk. I again thank Sajid for inviting me, and I thank IEEE Network for this opportunity. Thank you very much.